Okay, today we're going to get into details of the cell cycle. So the cell cycle is the series of events that occur as a cell grows and divides. And so the picture here can explain a lot. So this whole picture here is going to be the cell cycle. It's made up of two main parts. So we have interphase and we have the mitotic phase. Okay. The mitotic phase can also be called the M phase. Okay, so mitotic phase and M phase are going to be the same, where interphase is going to be the longest part of the cell cycle, and it consists of all of this. Okay, so it has three parts. Interphase has three parts. G1 is the first gap, and this is where growth is going to occur. So when the cell is going to start out really small, and it just is going to grow and get bigger. In the S phase, that's where the S stands for synthesis, so DNA replication is going to happen, and this is where we're going to double our DNA. And that's all the steps that we learned about before, and how DNA replication occurs with all those enzymes. That's happening in the S phase, which is part of interphase. All right, and then once that is done, then we go into G2, so the second gap phase. This is where more growth will occur, and this is also where preparation to divide, so it's going to prepare to divide. So if there's parts of the cell that need to be duplicated so that when the cell actually divides into two, it will have what it needs. To recap, interphase is going to be the longest phase of the cell cycle. It consists of three parts, G1, S, and G2. The DNA in interphase is going to be in chromatin form. Okay, so it's going to be thin, so if we wanted to look at it under the microscope, we would not be able to see the DNA because it would be so thin. Now, once the cell is going to divide, then it's going to be in the mitotic phase, which is also called, yes, the M phase, and that has two parts. That's going to have mitosis and cytokinesis. Mitosis is where we're actually going to divide the nucleus, and then cytokinesis is where we actually divide the cytoplasm, and we actually get two new cells. Each of those two new cells will then continue on with the cell cycle. Both of them will go into interphase, they'll continue to grow, then duplicate their DNA, and then go into G2, and then continue to divide if that's what the cells need to do. When we get into talking about the details of mitosis, there are a couple things that will be mentioned, and so we'll just get you familiar with them now. One is going to be the centrosomes. Centrosomes in animals consist of centrioles. So those are T-shaped noodles. And just so you know, I'll write it out here. So centrioles are the T-shaped noodles. And then they'll have little microtubules that will stick around. They kind of look like rays of sunshine around them. And the whole thing together, like that whole thing, is called a centrosome. And just know that the centrioles are only found in animals. But this whole region and like these little rays of sunshine will be in plants. And from those, they're going to send out spindles. So these spindles are just another form of microtubules. And they're going to attach eventually to chromosomes, okay, and kind of be pulling and pushing those chromatids and eventually chromosomes to itself. They'll be kind of moving them around. Okay, so spindles are these guys, the centrosomes are this whole thing, just so you know. Details of the cell cycle. So for interphase, here's our centrosomes, okay. We have a full nucleus, the DNA is all thin chromatin. It just looks like a normal cell that we studied in chapter 4 way long ago, okay. G1S, G2, all of that is in interphase. Once we hit mitosis, okay, the first phase of mitosis is going to be prophase. You'll notice the centrosomes are going to start moving away from each other. So they're moving away from each other. The spindles are starting to form. And the big thing here is that those chromosomes are actually becoming visible, okay? And again, don't forget they consist of two sister chromatids held together in the middle by a centromere. Okay, so under a microscope this might start to look like spaghetti that's forming inside of the nucleus. Your book talks about a prometaphase. Sometimes some books are just going to lump both of these phases together into one big prophase, so it's really long. Prophase, along with prometaphase, is considered the longest phase of mitosis because there's so much that's happening. And your book kind of breaks it up into just prophase and prometaphase. But in prometaphase, the nuclear membrane is going to start breaking apart. The best analogy I always heard was if you needed to shuffle a deck of cards, the first thing you'd have to do is take the cards out of the box, shuffle them around, and play your game. And so the nucleus is just the box. And so we have to get those chromosomes out of the box so we can shuffle and do the things that we need to do. So we get rid of the nuclear membrane. The nucleolus is going to go away. Those chromosomes are fully condensed. Spindle fibers, remember these little lines? They're going to start connecting at the centromere, which is just, so the kinetochore is just where those spindles attach at the centromere. Okay? All right, after prophase and prometaphase, then we have metaphase. And metaphase should be really easy, hopefully, to remember because those chromosomes are lined up in the middle in metaphase. They're going to be lined up in the equator. So if this is our north pole and this is our south pole, those chromosomes are lined up right there along the middle, which is the equator. Notice that the spindles are coming and they're going to attach, and that the chromatid, there's a chromatid facing like the north pole, and then there's another chromatid that's facing, towards the south pole, so that eventually, when we get over here to anaphase, they will separate. And so those spindles are going to pull the sister chromatid, which is now a single chromosome, or an individual chromosome, so it's in its own right, it's its own chromosome, and it's going to be moving all four of these guys up north, and these guys are all moving south. So for anaphase, they are moving apart from each other. Okay, like Anna is on the move. They're moving apart from each other. In telophase, it's going to be the opposite of prophase, right? So the nuclear membrane is going to be coming back. Those chromosomes are going to start unwinding into chromatin. The spindles are going away, and you'll start to notice that we're starting to pinch, which is the first part of cytokinesis. Okay, telophase, one way to remember it, perhaps, is that we're starting to get two, right? So we're starting to get two cells in telophase. And for sure in telophase we have two nuclei, so two nuclei in telophase. All right, cytokinesis is that second part of the mitotic phase, which is also known as the M phase. Again, mitosis divides the nucleus, and remember mitosis consists of PMAT, but it's going to divide the nucleus, where cytokinesis is going to divide the cytoplasm so that we can actually have two different cells. Usually it's going to happen while telophase is occurring. Cytokinesis is going to happen differently in plants and animals, other than the centrioles, which are only found in animal cells, and then how cytokinesis happens. Everything else is going to be the same between plants and animals, but cytokinesis is a big one. For plants, because this is animals, because animals are surrounded by a cell membrane, which is soft, it's almost like putting a rubber band around a balloon, and it's just going to contract and eventually get these two cells to actually divide and split. We call this a cleavage furrow. For G-rated purposes, I like to call it just a furrow. Okay, so animal cells create a furrow, and the direction of how this occurs is important. It starts on the outside and it moves its way in. Plants, on the other hand, have a really tough cell wall made of cellulose, and so it's really hard. You can't just take a rubber band and put it around a brick. That wouldn't make it divide in two. And so they need to have a different way to separate the cell. So what they do is they create a cell plate, which is just going to be depositing the cellulose along the inside until it reaches. So here's our picture. So here is the cell plate that's starting to form, and again the direction of it. And here is going to start from the inside and it's going to work its way out. Okay, so it's going to deposit cellulose along here. It's eventually going to fuse all of that together until eventually you actually get two separate cells. Okay? All right, I hope that helps. If you have any questions, feel free to look it over again. Other than that, have a great day, make good choices, and Jesus loves you.